All right, can everyone see that?
Yes, looks good.
Okay, great. So first, I just want to say thank you so much for the opportunity to talk about some of the work my lab has been doing using airborne eDNA techniques to track invasive forest pests, specifically in eastern hemlock forests. So in Michigan, eastern hemlocks are especially important because they serve as foundation species. So what that means is they have this unique ability to specifically regulate the environment in which they're found in. So their large canopy will regulate the amount of light that gets through to the forest floor. That can influence the temperature within a hemlock stand. They can also influence the soil chemistry and the amount of water yield that's held within that system. And because of this tight control that they have on this ecosystem, this can drive really unique biological communities within this forest setting. And for Michigan, preserving these hemlock forests is really important because we have over 170 million hemlocks within the state alone. However, these hemlocks are being threatened by a very tiny invasive forest pest known as hemlock woolly adelgid, also abbreviated as the HWA. So this is an invasive forest pest that's found within the eastern US and Canada. So you can kind of see it here on the left-hand side of your screen. This is a HWA crawler. So this is the most mobile stage of the individual. And these individuals are roughly less than a millimeter in size. So when HWA infests the hemlock tree, they feed on the hemlock sap and cause a decrease in photosynthesis, and this can result in hemlock mortality within 4 to 10 years. On the right-hand side of your screen is the distribution of HWA as up until 2022, and its relation to the native distribution of eastern hemlock. So the native distribution can be seen in the green, and then HWA's distribution can be seen in the brown. So the brown indicates areas that were infested prior to 2022. And then the yellow squares are counties that were infested or where infestations were detected in 2022. So in Michigan itself, we currently, before 2022, there were 5 counties currently infested, and then this infestation began in 2015, and this map is already out of date. We now have a current, infestation in Benzie County, so there are currently 6 counties that are infested. In Michigan, the majority of our surveying to detect HWA is through visual surveys. And so what happens is survey crews will go out and they will look for the presence of ovisacs. That's the middle picture where we have this white fluffy material known as HWA ovisacs that are present on the base of the hemlock needle. And so this is the primary mode of monitoring right now. And you can imagine with 170 million hemlock trees, going out and surveying for little white ovisacs in Michigan in the middle of winter can be a pretty daunting task. So we have been working with state and federal partners, trying to see whether or not we can use airborne eDNA techniques to help with this visual surveying that is conducted within the state. So airborne eDNA is just, like it says, eDNA that's collected from the air. So our lab has developed this 3D printed eDNA trap. So it has a base and a lid, and the base contains 4 microscope slides that are coated with a thin layer of petroleum jelly. We set these out in forested areas, and then our crews go out and collect the slides every other week, which we then bring back to the lab, remove all the petroleum jelly off of the slide, and then use quantitative PCR to see if we can detect HWA within these areas. And so prior to our current monitoring efforts, we needed to kind of look and see whether or not using airborne eDNA approaches would be a good method for HWA monitoring. So in 2021, we set out a study to kind of answer 2 questions. Number one, do our traps actually collect HWA or HWA-associated material like that ovisac material? And then number two, if it does collect HWA-associated material, how many traps would we actually need to deploy within a given area to have a high probability of capture success? So in 2021, we deployed 105 traps into a 90-acre area, which is outlined on the right-hand side. That 90-acre area was broken up into 5 replicate sections. And then within each one of those replicate sections, we deployed anywhere between 1 trap per 3-acre area up to 6 traps per 3-acre area to see what our density needed to be to have a probability of a detection of at least 0.9 or higher. So they were deployed from April to July, and with the slides exchanged every other week. And then we would bring back the slides back to the lab to see if any HWA was found. And so here are the results from that trap testing. Over the full deployment period, 96% of our traps captured either an adelgid or adelgid-associated material. And the traps that didn't collect anything were actually deployed in areas without any hemlocks, which kind of makes sense. And then on the right-hand side, we're looking at the number of traps that we would need to have a high probability of detection. So on the x-axis, we have our number of traps deployed per 3-acre area. And then on this y-axis, we have the number of traps that failed to detect anything, and the number of traps that succeeded in detecting HWA. And then we have on our other y-axis, the probability of capture success. And so from this, what we found, and here's our line of 0.9 probability of capture success. And what we found is ideally we would need 2 traps per 3 acres to have a probability of capture success of at least 0.9 or higher. When we talk to our partners, we recommend 1 trap per 1 acre to give a high probability of success. And so knowing our traps can at least capture something and knowing roughly what our density would need to be to have a high probability of capture success, we then got a grant from the Michigan Invasive Species Grant Program to use these traps to monitor for HWA in 2022 and 2023. So in these years, we deployed 55 traps across 11 sites. And these were in areas that our partners had designated as high-risk sites with no known infestations. These were deployed from the beginning of May until the end of July, with the slides exchanged every other week during that time period. We used HWA-specific primers to target a 250 base pair region of the cytochrome oxidase gene, and then we used quantitative PCR to test for the presence or absence of HWA. And this is an example of one of what our results would kind of look like for that presence absence detection. So we have a target threshold, that's a line that's kind of set here. Anything that's above that line would indicate a positive. Anything that is below that line would indicate either negative or non-conclusive. All of our samples are run in triplicate. And so for us, for something to indicate a positive, all 3 of those triplicates have to come back positive. So this would indicate 1 positive trap because all 3 triplicates came back positive. This would indicate a second positive trap. All of our traps all of our analyses are run with HWA positive controls to make sure our amplification is working. We do filled blanks, Vaseline blanks, and non-template control blanks as well to make sure that there's no contamination within our samples. And so from our 2022 and 2023 monitoring program, so this is data that's from one of our partner groups at Huron-Manistee National Forest. 6 of our traps tested positive for hemlock woolly adelgid in areas that they identified as high risks. So 2 of the traps tested positive in 2022. So these are those yellow triangles here. And then when they went to do visual assessment around these areas, they found 2 trees that were shown to be infested and have HWA ovocyte material, and those are indicated by the pink stars here. These trees were actually off of Huron-Manistee National Forest property. They were on private property, but they were very close enough that they actually treated trees within that property that were close to the area. In 2023, we had 4 traps that came back positive. So those are indicated by the red triangles here. And typically what happens is when we get a positive hit, we will contact our partners and we will say, we got a positive hit. Here are the GPS locations for the positive traps. And then when they conduct their surveys in the fall and winter, they can focus in on those areas. So this is relatively new data just from like a couple weeks ago, where they surveyed areas where we got positive hits. And here are the areas where they found infested trees. So there is one other positive trap that we have that we've indicated that's not on this map. It's in a different part of the forest. And they're going to be conducting visual surveys over the next couple of weeks in those areas. So in terms of management implications, we've found that using this type of approach is very effective, especially when you're trying to capture very small invasive forest pests that aren't able to easily be detected by other means. We find that it works very well, at least for our group, in tandem with visual surveys. So we get a positive detection and then we can direct survey crews to go out and visually assess in this area, because when you have 170 million hemlock trees, it's very hard to figure out where you want to visually look first. So this kind of points those crews in the right direction of, we think it's here, maybe check there first, and then you can kind of broaden your search area from there. And I think there are a couple of advantages for this type of technology. It's not just for HWA. So we know our traps can detect, or that can capture lots of different small insects or arthropods. We've done a little bit of amplicon sequencing to see what other type of things that we can detect on those traps. So this could be used not only to monitor for HWA, but potentially to monitor for multiple potential pests simultaneously. And so with that, I would just like to thank everyone in my lab who has been doing this work and going out and exchanging slides every other week for the past few years, and all of our partner groups.
Yeah, thank you so much, Charlyn. We do have one more speaker of the day. I will acknowledge we're a little bit over time, so if people have to leave for lunch, then I fully understand. And we will stay on just for a question period after our last speaker as well. So if you're able to stick around, please do so. So our last speaker of the day is Akli Zarouri. And yeah, whenever you're ready.
Good morning, everybody. Can you hear me?
Yes.
Okay. Okay. Thank you very much for having me. So my talk today is about our new technology dedicated to eDNA, which is integrated cartridge system for environmental DNA, which combine collection, extraction, and purification. This is the structure of my presentation. I will start by talking about some challenges related to eDNA methodology, and then I will describe our new technology, and then I will show some results of comparative performance testing between our kit, ICS kit, and Stevex Qiagen kit, which is currently the most used in eDNA. And then I will finish with the conclusion. What is eDNA? All living organisms release DNA into their environments, and we can use this DNA to determine these species. And this is the power of this method, because with one environmental sample, we can target many species at once. And it's also simple to implement. It needs collection, extraction, and purification, and then detection or quantification by using qPCR or sequencing. And the obtained data that are very valuable for invasive species management and bioconservation. Although this method is very powerful, but it still has some challenges to overcome. For example, the absence of standardization. In one hand, we have a wide range of filters, enclosed filters, open filters with different pore sizes. And in the other hand, we have many DNA extraction kits. This gives rise to multiple eDNA protocols, rendering comparison between different eDNA studies very difficult. During collection, we have an issue which can be encountered, which is filter clogging, and this limits the size, the sample size, which impacts the sample's representativeness. And we have also another challenge, which is in the presence of inhibitors, which are co-purified with eDNA that can inhibit qPCR and sequencing. And this is a big issue in itself, because it prevents the timely and early detection of invasive species. To overcome this, we designed a new technology, which is ICS cartridge. And here we have in the right the cross section of our cartridge, showing the assembly of different components. From the top to the bottom, we have a cap, and you have filter support and filter stabilizer, and in between we have we can insert many filters. Here, for example, there is gradient pore sizes, and then this device has two chambers. In chamber one, we can put the lysis buffer, which can lyse all the biological material captured in different filters. And then with one single centrifugation, we can harvest the whole lyse set into chamber two. And then by adding ethanol to chamber two and connecting the spin column, we can collect the whole DNA into the small spin column. To assess the efficiency of our new cartridge, we conducted a comparative study between ICS cartridge kit and the Stirbex Qiagen kit. This was side by side through 5 different parameters, which are workflow, filtration capacity, total eDNA yield, target common carp eDNA using nuclear and mitochondrial DNA. Knowing that common carp is considered as invasive species here in USA. And then we compare the probability of detection for both kits. Here is the comparative of workflow between Stirbex Qiagen kit in the top and our kit, ICS kit, in the bottom. So the workflow is pretty similar. So we have filtration and then extraction and purification. But there is some differences. The first difference, because the Stirbex Qiagen kit, it needs incubation at 56 degrees Celsius, which is not needed in our case. And the second difference is the need for the Stirbex cartridge to transfer the lyse set to the external tube, and then from the external tube to the spin column, Qiagen spin column. And this increases the risk of contamination. And this is avoided in our solution by directly connecting the spin column to the device. For the following steps, they are similar. We have 2 washing steps, and we have 1 elution step. Let's take a look to a comparative performance between ICS kit and the Stirbex Qiagen kit. Here we compare the volume capacity filtration. As one can see here, for ICS kit, we reached easily 2,150 milliliters and with stevas qiagen, we reached hardly 1700 milliliter. In terms of total DNA, ICS, with ICS we harvested 455 nanogram per microliter, and with Stevex qiagen we harvested 146 nanogram. So ICS harvested 3 times more eDNA than Stevex. Here we did a comparison between ICS and Stevex qiagen kit in terms of CT value. We targeted the nuclear eDNA of the common car. As we can see with ICS kit, we have a lower CT value with 32.1, and with Stevex qiagen kit, we have 34.72. Knowing that there is an inverse relationship between CT value and the amount of target DNA. So we estimated the difference fall between indicator, the amount of eDNA, which was estimated to 6-fold more captured with ICS cartridge than Stevex qiagen kit. In terms of probability of detection, we used here nuclear DNA. So the probability of detection with ICS removal, with ICS kit, we reached probability 100% of detection. With Stevex qiagen kit, we got just 10% of detection rate. After inhibitors removal, qiagen kit aligned with ICS kit. So this shows how ICS kit is resilient towards the qPCR inhibitors. For the probability of detection, we used also mitochondrial DNA. No, in this study, mitochondrial DNA was in minor amount. So the detection was enabled after inhibitor removal using Zymo inhibitor removal. As we can see here, the detection probability with ICS was 0.623, and for Stevex qiagen 0.2. With Stevex, the probability of detection cap was 3 times higher. With the ICS kit, was 3 times higher than Stevex. And this is very important for the early detection of invasive species if there are just a few individuals. So there is like just a small amount of DNA. In conclusion, I introduced the complete eDNA kit that combines filtration, extraction, and purification in one easy-to-use solution. And I showed that the ICS cartridge allows insertion of multiple filters with varying pore sizes, and these filters can be licensed in one step. And we saw that this cartridge is designed to avoid any cross-contamination by connecting the spin column directly to the cartridge. And we saw also that this kit is more resilient towards the qPCR inhibitors. All these features collectively contribute to its superior performance over the commonly used commercial kits in terms of volume filtration, total eDNA yield, which was 3 times higher, and target eDNA amount, which was 6 times higher, in terms of probability of detection of minor quantity of DNA, which was 3 times higher. And this is very important for timely and early detection of invasive species. Thank you for attention.
Okay, thank you so much, Akli. We will move on to our Q&A session now. So if all of our panelists are able to stay for a few more minutes, and we can address some of these questions in our Q&A box. I believe our first question here is for Chris Wilson, but Francine, you may also be able to answer this. Is the ministry investigating the application of eDNA for non-aquatic taxa? Could the same approach be applied in terrestrial environments? Maybe through passive air filtration, for example?
Short answer is yes. This was tested out for wild boar 2 years ago. There's a published assay, so it is available as an assessment tool. We've also tested this out for birds. The initial approach on birds was looking for endangered species, but both targeted and eDNA metabarcoding approaches work for terrestrial vertebrates. So the ministry has developed a number and then drawn others from the literature.
Great. Our next question, I believe, was for Francine and Rob. How accurate is eDNA ID? How do you ground truth the accuracy of the method?
I'm not sure if Francine's still here. And how accurate is it? I think Chris could field this question better than I could. Or there's Francine.
Yeah, I was going to say, I think Chris can answer that best.
Okay, and
Yeah.
for the record, there's others on here that could as well, like Caitlin and Charlene. So basically, if you're wanting to find just the species you're interested in, you need to test your assay against related species and other species that it could potentially co-occur with. So there's been quite a bit of effort globally put into this, and it's really, you're looking at a sequence of DNA that is unique to just that species. There's a lot of work that goes in for developing, testing, and validating. DNA barcoding gave the animal question a big leg up because there's a global database. What species you're interested in, look it up in the global barcoding database. It's called BOLD. And say, fine, I want to design primers that will amplify this and only this taxon. But it does require testing. Charlene or Caitlin, do you want to jump in on that?
Yeah. Yeah, I can say, at least for the primers that we use, we contacted the U.S. Forestry Service and pretty much said, send us every single adelgid species you have or DNA from it. And so, and then we tested it against every species, and we didn't get amplification for anything but HWA. And then I said, I don't believe that. And then I said, we tested it again. And so I think every few months I would start to doubt again, and then we would test it again. So I think probably after 4 rounds of my grad student just testing it over and over again, I started to believe it, and she was happy for that. But yeah, it's just a lot of testing.
So the next question I have is definitely for our Ontario panelists, and it kind of has been answered already, but we'll see if any of you have some insight in this question. Are there any recommendations for starting an eDNA project specifically looking for presence of zebra or quagga mussels within inland lakes? Who are some good contacts for this within Ontario, or are there any documents providing information?
So as far as I'm aware, there isn't a formal eDNA monitoring or surveillance program for either zebra or quagga mussels. And actually, Liz, you could answer this one. So the Invasive Species Center does have eDNA sampling kits available for stakeholder groups, but taking one sample off a dock for a lake and not detecting either Dreissenid mussel does not mean they're not there. So to do it, the important thing is look at sampling design. Where would you want to look if you think they're likely to come in? So I would say for privately owned water bodies or shorelines around them, the ISC would be the point of contact. The province does have assays for these, and there's a number of labs across the country and continent that will do this testing. But the MNRF right now does not have a monitoring program in place, just the tools.
Yeah, so my coworker there, Lauren, has dropped our informational email into the chat. So yeah, the Invasive Species Center is a great point of contact for that. We do have eDNA sampling kits available for community members, and we can absolutely try to collaborate on that if that's something that you would be interested in. This question, I believe, was for Caitlin, but it's kind of a general question as well. Can you explain why you do the PCR replicates? What is different about each PCR test, and does each PCR test for a different gene sequence?
Yeah, so each PCR reaction for a single sample acts as another point of replicate. Can you still hear me?
Yes.
Okay, sorry, I thought I froze. So that just allows us to have more chances to detect the eDNA because one, the pythons secrete less, so we have very, very low concentrations of eDNA present in our sample. And when you mix that into like a volume of something, that low concentration is going to be very sporadically placed within that sample. And so allowing us to run more replicates in the PCR allows us to test more and more instances where that random eDNA copy could then be detected in the PCR. This really just tests like if eDNA is present in the sample, rather than just running one and saying no or yes. We run 5 times 5 to see if it's no or yes for the geographic site. So that's just more instances for us to detect eDNA. It's not a different gene. It's not a different assay that's testing anything. It's the same thing across the board. It's just doing the PCR reaction essentially 25 times to see if eDNA is detectable in that geographic area. And we split it by sites, we set it, or samples, I'm sorry, we split it by samples and PCR reactions just to give us more instances of that.
Okay, thank you so much, Caitlin. I think, there's a ton of other questions left, but I think unfortunately we're going to have to end our question period for today just because we're bleeding into lunch quite a bit. So that's all the time we have today for questions. Again, please reach out to our speakers if you have unanswered questions. I just wanted to give a quick reminder about a trivia event happening on Thursday, February 15th at 11:45 a.m. This is a chance for you to showcase your invasive species knowledge for the chance to win a prize. All you need to do is join the trivia session and Zoom events and have your mobile device handy. And I wanted to personally thank all of our amazing speakers today for carving out the time to spread invasive species awareness and knowledge. I hope everyone tunes in for the rest of the day's events, and I believe Maddie is closing us out with a few extra reminders.
Yes, thank you so much, Liz, and thank you to all of our wonderful speakers and attendees. We're so sorry for cutting lunch short here, but hoping you can grab a quick little something to bring back to your screen so you can join us at 12:30 for our first keynote speaker with Henry Lickers, who will be delivering our presentation titled May We Live in Interesting Time, Indigenous View. That'd be one you won't want to miss, so hope to see you then. Thank you all so much.